It is estimated that the human body contains about 37.5 trillion cells, with a total of 6 billion base pairs of DNA per cell. The human genome, if stretched out end to end, would be 1.8 meters in length. So each of us has enough DNA to reach from here to the sun and back more than 300 times. How could this be possible? In order to fit the whole genome into a cell nucleus at the micrometer scale, DNA must be packaged and coiled tightly. Histone proteins are a family of small, positively charged proteins which facilitate coiling of the DNA, binding tightly due to DNA's negatively charged phosphate backbone. The unit of chromatin containing DNA and histones is called the nucleosome. It consists of a core of 8 histone proteins and about 146 base pairs of DNA wrapped around almost 2 times. To form the nucleosome, first histones H3 and H4 make dimers. Histones H2A and H2B also make dimers. The H3-H4 dimers then come together to form an H3-H4 tetramer which binds to DNA. The DNA tetramer complex combines with 2 H2A-H2B dimers, resulting in the DNA octamer complex, the nucleosome. Each chromosome is thus a long chain of nucleosomes which looks like a string of beads when viewed using an electron microscope, called the 11 nanometer fiber or euchromatin. DNA in this form is accessible enough for transcription. The histone protein H1 binds to another 20 base pairs of DNA as well as the nucleosome core. The histones' N-terminal tails also interact with the DNA. Thus, the histones condense the linker DNA to form a structure called heterochromatin, or the 30 nanometer fiber. This is a supercoiled structure that can be represented by a winding telephone cable, showing how tightly compacted the DNA can get. It is unknown whether this supercoiling forms a solenoid structure with 1 helix or a zigzag structure with 2 helices. Transcription factors are sterically unable to bind the DNA, and it takes histone-modifying enzymes changing the histone tails, such as acetyl, methyl, and phosphate groups, to change these interactions and thus decondense the structure. Further packaging is only found during cell division, predominantly metaphase, as the DNA must be segregated into daughter cells. This involves the next level of DNA packaging, where loops of 30 nanometer fiber supercoil around SMC proteins. Further loops and condensation produce the neatly packaged mitotic chromosome.